So, the neurons get an awful lot of attention in neuroscience and have for a very long time because, they're wire-like, right? They have, clear integrating, inputs here. They can, these are synaptic connections that are made, on the dendrites, for example, up to 10,000 per individual neuron that can be excitatory, and we're going to see that'll, actually depolarize, the term we'll use soon. It'll push the neuron towards initiating an action potential, an electric current that'll run down its length to the end here, and provoke the release of neurotransmitter, into a synaptic gap or cleft that could influence the next cell. It could also be inhibitory. We'll talk about the fancy term for that it's going to be hyperpolarizing. All these terms will make more sense soon. But there's like a lot of integration occurring here at the, the dendrites, maybe on the cell body to some extent, a decision being made here at a little bulge at the very beginning of the axon called the axon hillock. And then, the sort of the flow of current, basically, the action potential that'll work its way down the axon to its end. But there are, 86 billion of these cells. Their electrical transmission and chemical communication at synapses made, neurons really the focus of interest in terms of information processing in the brain. However, there's a vast number of additional cells in our nervous systems. They're called glial cells. And glia, is a Greek word for glue. And, initially they were thought, they're the cells that are just like literally the support cells, only support. They just provide support, for the stars of the brain, right, which are the neurons. But we're learning more and more that glia are absolutely essential for brain function, for brain development, and also for information processing as well. So they carry out a number of very, very critical functions and are on par, really, with the neurons. So neurons and glia are the cells of the nervous system that we need to know if we're going to be studying neurophysiological psychology. There are three primary classes of glial cells. Two of these are referred to as macroglia. That means big glia, the big glia. These are the astrocytes. And a set of glial cells that are called the myelinating glia. And then there's another, group of glia. They're called microglia. These are the smaller, much smaller glia. They're going to actually be much more mobile, we'll see, and also derived from a different embryonic layer. That's, also the source and origin of, cells of the immune system and the rest of the body. So the two macroglia, the big glia, one is known as the astrocyte. And astro, of course, means star. So this is like the star-shaped glial cells. They wrap their membranes, their phospholipid bilayers, around, points of synaptic connection. So, where one neuron meets another, this particular, point-to-point, chemical gap here, or a gap that's, bridged by the release of a neurotransmitter from the first neuron, which will then, act upon typically, the membrane of the next neuron, is actually wrapped. There's some integrity, about that chemical communication there, partly because the astrocyte wraps portions of its membrane around, that synaptic connection. And also other projections of these star-shaped astrocytes, these macroglia, wrap around the thin capillaries that work their way through, all different portions of the brain, and spinal cord and nervous system. So if you can imagine, like these astrocytes are intimately associated with synaptic connections. They're wrapping both the synaptic, link, and then also wrapping the capillaries. And what we found is that they do a number of things. One, they contribute to the blood-brain barrier because astrocytes release chemicals that will, be detected by, received by the cells that form the walls of the capillaries, the plumbing, the tubes themselves that carry the blood, and it'll provoke this kind of tighter adhesion of the membranes of these capillary cells, to each other to make the plumbing in the brain less leaky. So the astrocytes contribute directly to the development and maintenance of a blood-brain barrier. In addition, they maintain that point-to-point chemical connectivity, where the neurotransmitter is released from one neuron and received by the next, partly because there's a membrane, wrapping around that synaptic connection, which prevents the sort of loose diffusion, widespread diffusion of the neurotransmitter. So it keeps it, kind of close. But also astrocytes have receptors. These we're going to see are proteins that are embedded in membranes that can receive or bind, chemicals, including neurotransmitter that's released by the presynaptic terminal, at a synapse. And so they can measure to some extent. We're going to see the level of synaptic activity that's occurring at a particular synapse. How much neurotransmitter is being released? How often are these receptors stimulated or receiving neurotransmitter messages? And they link that detection of enhanced transmitter activity, if there's a lot of synaptic activity occurring, to influence over those capillaries. They actually release chemicals in response to lots of synaptic activity that'll provoke the capillary to expand, to actually allow for more blood flow into a particular area of the brain. And this is important because you want, neurons that are active to get fed, so there's going to be enhanced blood flow into areas where there's greater, neuronal synaptic activity. And we're going to see that's actually the basis for many brain imaging techniques like fMRI and PET. In addition to the astrocytes, the other big class of macroglia are called the myelinating glia. And myelinating glia wrap their phospholipid bilayers, their membranes, around segments of a neuron's wire-like axon. So these little white segments here are actually membrane that's contributed by, glial cells, the myelinating glia. In the central nervous system, we have what are known as oligodendrocytes. They're the myelinating glia that you find in the brain and spinal cord. But in the periphery, you actually have myelinating glia that are called Schwann cells, which are named after a guy named Schwann, that wrap their membranes around the membrane of the axon, of neuronal axons. Now, the phospholipid bilayer that's contributed by the myelinating glia, the oligodendrocytes in the central nervous system and the Schwann cells in the periphery, along with proteins that are actually embedded in that phospholipid bilayer, because the glial cells will actually express genes that produce proteins that get embedded in their membrane that wraps around. So these are called myelin-associated proteins, or MAPs. This is actually the membrane itself, and those proteins are known as myelin. It's basically phospholipid bilayer, basically, of the myelinating glia that wrap around segments of axon. And myelin actually will speed up electric current flow along an axon, during an action potential, and we'll discuss how this is accomplished, pretty soon. There are other differences between the oligodendrocytes, which are the myelinating glia in the central nervous system, and the Schwann cells, which are the myelinating glia in the periphery. The oligodendrocytes actually release chemicals that restrict neuron growth in the brain and spinal cord, while growth factors are actually released by Schwann cells in the body. And I'm just curious what you'd think about that. Like, why would there be a difference? Why would you be inhibiting, neuronal growth in the CNS while potentially encouraging, neuronal regrowth and repair in the peripheral nervous system? And finally, the last, form of glial cell, the third class of glia, are called the microglia, and they're much smaller than the macroglia, which again are the astrocytes and the myelinating glia. They are CNS representatives of the body's immune system, and they are exceedingly mobile. So they move like little spiders throughout your brain and spinal cord. They literally touch synaptic connections with their projections, their spider-like projections, and occasionally they will morph into what's known as the activated form. They change physically, actually, and they can act to remove errant synapses, like synapses that appear to upset them in some way. and or microorganisms like, bacteria or something, or some kind of an infection that might be invading the brain. So the glial cells are, just rather fascinating and critical components of our nervous system, in addition, to the neurons, that have for, many years been sort of the star of the